Good afternoon. Can you hear? Good afternoon. Can you hear? Jagruti, can you hear? Bhavini? ऐकू येते का? शिवानी? Neha? Okay. So let us start with the last point, that is HSP and molecular chaperons. So what are these HSP? HSP are heat shock proteins. You have studied extremophiles in the first semester. So these extremophiles can tolerate the extreme conditions because of special type of proteins known as heat shock proteins. So these HSP are a family of proteins that are produced in response to exposure to stressful conditions. So if the organism is exposed to some adverse conditions or stress like conditions like extreme alkaline, extreme acidic condition, low pH, or high pressure, high temperature, low temperature, psychrophilic habitat, then in that conditions those HSPs are activated. So whether they were first described in response to heat shock, so they were given the name HSP, but that doesn't mean that these proteins are present only in thermophilic organisms or organisms exposed to high temperature. They may be produced by organisms exposed to cold, to ultraviolet light, or they may also be activated when the organism is damaged and it is undergoing repair mechanisms. तर त्यावेळी सुद्धा हे HSPs ॲक्टिवेट होत असतात. Many members of this group perform chaperone functions. So chaperone function is nothing but stabilizing the newly synthesized proteins. So that is known as actually the chaperone function. Most of these heat shock proteins, they play the role of chaperones. That is, they stabilize the new proteins to ensure the correct folding of these proteins, or they refold the proteins that were damaged by stress conditions, or if the cell is exposed to stress conditions, then these heat shock proteins, stress conditions मध्ये normal proteins may undergo some type of damage. In that case, chaperones function helps to stabilize those proteins, and this chaperone function is carried out by some heat shock proteins. So increase in the expression is transcriptionally regulated. So that is, more and more chaperones or HSPs will be synthesized only when the stressful condition arises, and that is controlled by the process of transcription. So upregulation of heat shock proteins is the main part of heat shock response. That is, more and more synthesis of heat shock proteins will take place when there is exposure to some heat shock conditions, and they are present in virtually all organisms including bacteria to human beings. They are named according to their molecular weight. For example, HSP 60, HSP 70, HSP 90. So 60, 70, 90 kilodaltons is the molecular weight of these heat shock proteins, and they are written as HSP60 so that we can differentiate between them. सगळे हीट शॉक प्रोटीन्स आहेत, पण त्यांना डिफरन्शिएट करण्यासाठी आपण त्यांना हे 60, 70, 90 त्यांच्या मॉलिक्युलर वेटचं नंबर तिथे ऍड करत असतो. The small 8 kilo Dalton protein ubiquitin, which marks proteins for degradation, has also the features of heat shock protein. आपण ubiquitin बघितलेला आहे. So it's a very small 8 kilo Dalton protein, and some of the properties of this ubiquitin resemble the properties of heat shock protein. म्हणून आपण ubiquitinला सुद्धा heat shock protein मध्ये कन्सिडर करू शकतो. आणि ubiquitin is the main protein involved in tagging the proteins which are to be degraded, the proteins which are of no use, the proteins which are uh, like incomplete proteins or proteins which are not biologically sound. These proteins need to be degraded and they are tagged by ubiquitin. This protein also shows heat shock like properties. So a conserved protein binding domain of 80 amino acid alpha crystallins are known as small heat shock proteins. सो हीट शॉक प्रोटीन्समध्ये पण SHSPs असतात आणि these are nothing but the proteins which have 8 amino acids alpha crystallins. So they are very very small chains, polypeptide chains, फक्त 80 amino acids चे small chains असतात आणि they are present in alpha स्ट्रक्चर, म्हणजे अल्फा चेन बीटा चेन आपण जी म्हणतो, तर त्या अल्फा चेन फॉर्म मध्ये ते प्रेझेंट असतात, अँड दोज आर नोन ॲज स्मॉल हीट शॉक प्रोटीन्स. व्हॉट इज द रोल ऑफ HSPs? So upregulation in stress is the main function of HSPs. So that is, under stress conditions they are synthesized. So high levels of heat shock proteins have been like एक्सपेरिमेंटल कंडिशन्स देऊन तुम्ही त्यांना वेगवेगळे शॉक ट्रीटमेंट्स देऊ शकतात. हीट शॉक म्हणजे फक्त हीटच नसतं, तर केमिकल्स असू शकतात, अल्ट्रावायलेट रेडिएशन्स असतात. तर यू कॅन expose the organisms and you can stimulate the production of heat shock proteins by exposing these organisms to stressful conditions. So such type of stressful conditions may include exposure to toxins किंवा एखाद्या बॉडीमध्ये किंवा एखाद्या एक्सपेरिमेंटल ॲनिमलमध्ये इन्फ्लमेशन झालेलं असेल, सूज आलेली असेल, इन्फेक्शन झालेलं असेल किंवा ते टॉक्सिन्सला एक्सपोज झालेले असतील, लाईक इथेनॉल, आर्सेनिक, ट्रेस मेटल्स किंवा अल्ट्राव्हायलेटला एक्सपोज झाले असतील किंवा काही सेल्सला आपण स्टार्वेशनला फेस करायला लावलं असेल, त्यांना न्यूट्रिएंट्स नाही सप्लाय केले असतील किंवा ऑक्सिजन कमी केलं असेल, त्यांचं अनरोबिक कंडिशन्स क्रिएट केल्या असतील ज्याला आपण हायपॉक्सिया म्हणतो, किंवा प्लांट सेल्समध्ये नायट्रोजन डेफिशियन्सी केली असेल, म्हणजे प्लांट्सला नायट्रोजन प्रोव्हाइड नसेल केलं किंवा पाणी दिलं नसेल, तर हे सगळ्या कंडिशन्सला आपण ॲडवर्स कंडिशन्स म्हणतो. अँड इफ यू वॉन्ट टू स्टडी HSP, you can expose these cells or organisms or plants or animals to such type of conditions and study what types of protein synthesis is stimulated in such organisms, आणि त्याच्यावरून तुम्हाला HSPचा अभ्यास करता येऊ शकतो. The mechanism by which HSP activates the heat shock factor has been determined in bacteria. तर बॅक्टेरियामध्ये इट्स व्हेरी सिम्पल. त्यांना ॲडवर्स कंडिशन्सला एक्सपोज करून त्या सेल्समध्ये म्हणजे तुम्ही सॅम्पल घेऊ शकतात आणि कुठले प्रोटीन्स आहेत, प्रोटीन प्रोफाईल त्याचं स्टडी करू शकतो. तुम्हाला जर आठवत असेल तर मागच्या वर्षी एक पुणेहून सर आलेले होते आणि त्यांनी माल्टीटॉफ इन्स्ट्रुमेंटचं तुम्हाला एक लेक्चर दिलेलं होतं. ओके, सो ते माल्टीटॉफ विल गिव्ह यू द प्रोटीन प्रोफाईल ऑफ एन ऑर्गॅनिझम. तर माल्टीटॉफच्या मदतीने तुम्ही नॉर्मल ऑर्गॅनिझम्सचा प्रोटीन प्रोफाईल काढायचा, मग त्याच ऑर्गॅनिझम्सला तुम्ही स्ट्रेस कंडिशन्सला एक्सपोज करायचं आणि परत त्याचा प्रोटीन प्रोफाईल काढायचा. दोघं प्रोफाईल कंपेअर केल्यानंतर तुमच्या लक्षात येईल की काय डिफरन्सेस आहेत. डेफिनेटली त्यांना स्ट्रेस कंडिशन्सला एक्सपोज केल्यानंतर यू विल सी द प्रेझन्स ऑफ सर्टन HSPs synthesized in the organisms exposed to stress. So here you can use such instruments and they can give you very good results. So असे प्रोफाईल असे टाईपचे प्रोजेक्ट्स पण खूप बेनिफिशियल असू शकतात. तुम्ही MSc लेवलला असे प्रोजेक्ट्स करू शकतात. बट अनफॉर्च्युनेटली you are not in a position to do any projects, and there is no hope that you will do such projects in the fourth semester also, because nothing is uh, seen fruitful. Everything is going in adverse directions. तर या ॲडवर्स कंडिशन्समध्ये आपल्याला डाऊटफुल आहे की तुम्हाला प्रोजेक्ट करायला मिळेल की नाही. Now, during heat stress, outer membrane proteins do not fold and cannot insert correctly into the outer membrane. सो असे जे प्रोटीन्स आहेत ते प्रॉपरली आउटर मेम्ब्रेनमध्ये इन्सर्ट होऊ शकत नाही जेव्हा हीट स्ट्रेस असेल. दे एक्युमुलेट इन द पेरिप्लाझ्मिक स्पेस, द स्पेस बिटवीन द cell wall and the cell membranes. These outer membrane proteins are detected by DEG-S, an inner membrane protease that passes the signal to the membrane through the membrane to the sigma E transcription factor. अशांना डिटेक्ट करण्यासाठी परत सिग्नल प्रोटीन्स आहेत, ज्याला आपण इनर मेम्ब्रेन प्रोटीएज आहे तो ते सिग्नल पास करतो टू सिग्मा ई ट्रान्सक्रिप्शन फॅक्टर्स. आणि मग आपल्याला लक्षात येईल की ह्या स्ट्रेसफुल कंडिशनमध्ये पहिला इफेक्ट तुम्हाला काय दिसेल, फिनोटायपिक इफेक्ट, की ते प्रोटीन्स आउटर मेम्ब्रेन प्रोटीन असून सुद्धा आउटर मेम्ब्रेनला बाइंड होत नाही, तर ते पेरिप्लाझ्मिक स्पेसमध्येच दडलेले राहतात. सो इन दॅट केस द सिग्नल विल बी गिवन बाय DEG-S. So they will be detected. Such type of outer membrane proteins in the periplasmic space can be detected by some other protease, that is DEG-S protease, and it will pass the signal through the membrane to the sigma E transcription factor. So the sigma factor is mainly involved in the initiation of ट्रान्सक्रिप्शन. तर त्याला जे सिग्नल मिळेल त्यानुसार मग HSP सिंथसाइज होऊ शकतील. Some such studies suggest that increased in damage or abnormal proteins bring HSP into action. Many times, signal will be given if the proteins are damaged due to stressful conditions. So, stressful conditions मध्ये normal proteins जर डॅमेज झाले तर ते डॅमेज प्रोटीन सुद्धा अशाच पद्धतीने सिग्नल देऊ शकतात to the sigma factors, and these sigma factors will then initiate the process of transcription. You know very well sigma factor कुठे असतो, RNA polymerase ला bind होतो. Unless and until sigma factor binds to RNA polymerase, the holoenzyme will not be synthesized. RNA polymerase will remain inactive. The holoenzyme synthesis साठी sigma factor is essential, and that is why we call it as the factor essential for initiation of transcription. So when sigma factor binds to the RNA polymerase will be able to bind to the promoter and start the process of transcription. So abnormal proteins can give the signal to sigma factor, whereby sigma factor can bind to the RNA polymerase, form the holoenzyme and start transcription. Second role as chaperone. So several heat shock proteins, they function as intracellular chaperones for other proteins. Chaperones are proteins which help in stabilization or folding or unfolding or interactions. So they play an important role in protein-protein interactions such as folding. Whereas where at rest the proteins are present in a folded form. So for this folding, interactions between certain amino acids will be essential. किंवा वेगवेगळे टाईपच्या चेन्स असतील प्रोटीन्सच्या, त्या बाइंड होऊन राहायला पाहिजे. तर त्या प्रोटीन इंटरॅक्शन्स घडवून आणण्यासाठी heat shock proteins play the role of chaperones. They help in the establishment of proper protein conformation and prevention of unwanted protein aggregation. So they will also not allow aggregation, unwanted aggregation of different types of proteins. So wherever aggregation is not required, they will separate the proteins. Wherever folding is required, they will fold the proteins, and this particular role is known as the role of a chaperone by helping to stabilize partially unfolded proteins. So if the proteins are partially unfolded during synthesis, those will be stabilized by the HSPs. They help in transporting proteins across the membrane within the cell. म्हणजे असे पार्शली अनफोल्डेड प्रोटीन्स असतील तर ते अनस्टेबल असतील तर त्यांना सेफली मेम्ब्रेन मधून पास करून सेलपर्यंत पोहोचवण्याचं काम सुद्धा ते करतील. म्हणजे सपोर्टिंग रोल विल बी प्लेड. That role is known as the role of a chaperone. Not only heat shock proteins will give signals, okay, but chaperones will give the supporting role. Second, management. Heat shock proteins फक्त स्ट्रेसफुल कंडिशन्स मध्ये सिंथसाइज होतात का? नो. They may also be present in non-stressful conditions, and during that non-stressful condition they will play the role of monitoring. Okay, so they will only monitor the cell's proteins. Are they normal? Are they old? Are they new? Are they should these proteins they which have no use, or if they have become old proteins, should they be degraded by proteasome? So that role is known as monitoring, and by monitoring they will carry the old proteins to the recycle bin, that is proteasome. So this recycle bin मध्ये गेल्यानंतर 19S cap of the proteasome will take in that old protein, degrade it into the peptides or the amino acids. These activities are the part of cell's own repair system called the cellular stress response or heat shock response. So this management ही प्रत्येक सेलची इनहेरंट ability आहे, and this inherent ability of the cell to repair its own system is known as, means removal of old proteins, synthesis of new proteins. This is known as the management system, and that is known as cellular stress response or heat shock response. Then they have these HSPs, they also have cardiovascular role. They serve a significant cardiovascular role like HSP 90, HSP 84, 70, 27, 20, alpha beta crystalline, all have been reported to play a role in cardiovasculature. म्हणजे to control the relaxation, contraction of the muscles of the heart. HSP90 binds both endothelial nitric acid synthase and soluble guanylate synthase, cyclase, which in turn are involved in vascular relaxation. So what these heat shock proteins do, they bind to certain chemicals or enzymes, and these enzymes are mainly playing the role in relaxation of the vascular muscles of the heart. So those enzymes are nitric oxide synthase and soluble guanylate cyclase. ह्या दोघी एन्झाईम्सला ते हीट शॉक प्रोटीन्स बाइंड होतात आणि अशा पद्धतीने वॅस्क्युलर मसल्सचं रिलॅक्सेशन शक्य होऊ शकतं. अनलेस अँड अंटिल हार्ट मसल्स रिलॅक्स and contract, breathing is not possible. Next role is in immunity. So this is a very important role of HSPs. They are playing a very significant role in immunity, not as a whole protein, but also in the form of peptides. Means HSP as a whole नाही, तर HSP चे छोटे छोटे पेप्टाइड्स, त्या प्रोटीनचे छोटे छोटे पेप्टाइड्स सुद्धा इम्युनिटी मध्ये खूप इम्पॉर्टंट असतात. The affinity and specificity of this interaction is low. It was shown that some HSPs possess the ability, like HSP 70, 90, 96, calcrate, calreticulin, and their peptide binding sites were identified. तर ह्यांच्यामध्ये या हीट शॉक प्रोटीन्समध्ये ती पेप्टाइड बाइंडिंग साइट्स दिसून आलेले आहेत जे इम्युनिटीमध्ये युजफुल असतात. For example, in case of GP96, it is not clear whether it can bind to peptides in vivo in living conditions. पण in vitro मात्र GP96 पेप्टाइड्सला बाइंड होऊ शकतो. And peptide binding sites have been reported in GP96. But GP96 immune function could be peptide independent. असंही शक्य आहे. हे गरजेचं नाही की ते पेप्टाइड्सला बाइंड होतात. Peptides means what? Yes, antigen, any type of antigen. If it is coming there in the form of a protein, these heat shock proteins can bind to them just like immunoglobulins. So GP96 doesn't show this peptide binding, but uh, its immune activity must be peptide independent. It is involved in the folding of immune receptors like PLR or integrins. All these are involved in immunity. Next is molecular chaperons. So in molecular biology, molecular chaperons are nothing but the proteins which help in folding, unfolding, assembly, disassembly of various structures, uh, and thus they help in maintaining the conformation of a particular macromolecular structure. They are present where macromolecules perform their normal biological functions. So HSPs will be synthesized in response to stress, but chaperones, they will be present always, and they usually are found in the regions where the proteins are performing normal biological functions. So there they have been reported to play the role of assembly, folding, etc. The main role is protein folding. The first protein to be called a chaperon assisted the assembly of nucleosomes from folded histones and DNA. So first type of chaperon was found involved in folding of uh, nucleosomes. So they are found in the assembly of nucleosomes, and where these were found in the DNA histone complexes. And these DNA histone complexes were found in the nucleus of eukaryotic organisms. So there they were reported to play the role of folding. One major function of chaperone is to prevent both newly synthesized polypeptide chains and assemble subunits from aggregating into non-functional structures. नवीन नवीन जे प्रोटीन्स सिंथसाइज होतात ते चुकीच्या पद्धतीने they should not be assembled. Their assembly should be the correct assembly. They should not form aggregates which are of no use. And in that case, chaperones will not allow the aggregation of proteins which are non-functional or which are not useful. So that is why chaperones uh, cannot be called as heat shock proteins, because of the tendency to aggregate increases as proteins are denatured by stress. So heat shock proteins, on the contrary, like chaperones, they will be activated whenever the folding, unfolding, assembly, aggregation is required. But HSP, they cannot, the tendency to aggregate increases. जे प्रोटीन्सला ते ॲग्रिगेट करतात, जेव्हाही ते प्रोटीन्स स्ट्रेसमुळे डिनेचर होतात तर त्यांना ॲग्रिगेशन करायचं काम एच एस पीचं असतं. आणि हेच शॅफेरॉन्सचं काम काय आहे की ते अननेसेसरी ॲग्रिगेशन अवॉइड करतात. Chaperones do not contain any additional steric information required for the proteins to fold. However, some specific steric chaperones do convey unique structural information onto proteins. They don't have that particular information to carry out the folding, but they play a supporting role. त्यांच्यामध्ये स्वतःमध्ये असं काहीच नसतं. But still, certain steric chaperones, means those which are involved in the conformation, they can show this particular characteristics, and they can convey this message to the proteins for aggregation or disaggregation or folding. Such proteins violate Anfinson's dogma requiring protein dynamics to fold correctly. So let us see Anfinson's dogma म्हणजे the ability of the protein to fold correctly. तिथे हे शॅफेरॉन्सचा मेन रोल असतो. तर ते मात्र हे स्टेरिक शॅफेरॉन्स मात्र ते रूल फॉलो करत नाही. Examples of some chaperons, HSP60, it is known as GROW-EL, GROW-ES complex in E. coli. It is 1 million Dalton chaperon complex. GROW-EL is a double ring, 14 units containing hydrophobic patch, म्हणून त्याला 14 मर असं म्हटलेलं आहे. It is so large that it can accommodate native folding of 54 kilodalton GFP in its lumen. तर त्या प्रोटीनचं फोल्डिंग जे 54 किलो डाल्टनचं प्रोटीन आहे त्याच्या फोल्डिंगला ते सपोर्ट करू शकतो. ग्रो ES इज अ सिंगल रिंग हॅप्टामर, 7 युनिट्स असतात त्याच्यामध्ये, दॅट बाइंड टू दी ग्रो EL इन प्रेझन्स ऑफ ATP अँड ADP. सो हा जो कॉम्प्लेक्स आहे ग्रो EL अँड ग्रो ES, so that complex carries out the HSP role, or they can also carry out the role of a chaperone. So they may not be able to undergo previous aggregation, but it does compete in the pathway of misfolding and aggregation. So they are playing the role of a molecular chaperone. Next is HSP70. It is known as DNA-K in E. coli, and it is the best characterized small chaperons of only 70 kilodaltons. It looks like this. This is the image of HSP pocket. इथे असं pocket like structure आहे. Okay, so that is HSP70. The HSP70 proteins are helped, they help the HSP40, or they are helped by HSP40 proteins, that is DNA J in E. coli. E. coli आणि eukaryotes मध्ये HSP ची नावं वेगवेगळी आहेत. They increase the ATP consumption rate and activity of HSP70. So HSP40 plays a supporting role for HSP70. And what is that supporting role? It helps in increasing the ATP consumption rate, that is energy consumption rate. This helps in increased expression of heat shock protein 70, and that is involved in apoptosis, that is death of the old cells, unwanted cells or unwanted proteins. Although a precise mechanistic understanding has yet to be determined, it is known that HSP70 have a high affinity bound state to unfolded proteins when bound to ADP, and low affinity state when it is bound to ATP. म्हणजे जेव्हा ATPचं कन्व्हर्शन ADP मध्ये होतं तर इट गेट्स एनर्जी, अँड ॲट दॅट टाइम ADP फॉर्ममध्ये जेव्हा ATP असतो म्हणजे ॲडिनोसिन डायफॉस्फेट, तेव्हा ती एनर्जी रिलीज झालेली असते. त्यावेळी ती एनर्जी ॲब्सॉर्ब करून HSP70 ती एनर्जी ॲब्सॉर्ब करण्यासाठी HSP40 त्याला मदत करतो, आणि HSP70 कम्स इन हाय अफिनिटी bound state, means it will be having affinity to show its action. And whenever ATP फॉर्म मध्ये ATP प्रेझेंट असेल तेव्हा ती एनर्जी रिलीज झालेली नसेल तर HSP will be in low affinity state. It is thought that many HSP70 crowd around the अनफोल्डेड सबस्ट्रेट, म्हणजे प्रोटीन जेव्हा अनफोल्डेड फॉर्ममध्ये असतो तिथे खूप HSP कलेक्ट होतात, अँड ते स्टॅबिलाइज इट, ते प्रिव्हेंट दी एग्रिगेशन अंटिल दी अनफोल्डेड मॉलिक्यूल फोल्ड्स प्रॉपरली. म्हणजे प्रॉपर फोल्डिंग जिथे नसेल किंवा अनफोल्डेड स्टेटमध्ये प्रोटीन्स असतील तिथे हे HSP प्रोटीन्स कलेक्ट होऊन जातात आणि ते तसेच तिथे राहतात जोपर्यंत द करेक्ट फोल्डिंग डझंट टेक प्लेस. So when the correct folding takes place, HSP70 proteins, they lose the affinity for the that particular substrate and they go away from the substrate. HSP70 also acts as a mitochondrial chloroplastic molecular chaperone in eukaryotes. So they play a significant role in mitochondrial proteins as well as chloroplast proteins, and there they play the role of chaperones or they help in stabilization of mitochondria and chloroplast. Next one is HSP19. It is known as TPG in E. coli. It is a very less understood chaperone or heat shock protein. 90 kilodalton is its molecular weight, and it is found to be responsible for viability in eukaryotes. Sometimes even in prokaryotes also they are responsible for the viability for living. HSP90 is a molecular chaperone essential for activating many signaling proteins in eukaryotic cells. So they are useful in signal pathways or signal transduction. It has an ATP binding domain, a middle domain, and a dimerization domain. So ATP is essential for its activity. It may require co-chaperones like immunophilins, STI1, P50, AHA1, and also it sometimes cooperates with HSP70 chaperone system. So individually तो काम करण्यापेक्षा it binds to certain other chaperones and then it shows its action. So those chaperones are, co-chaperones are immunophilins, STI1, P50, CDE, CDC37 in case of E. coli, AHA1 आणि अशा chaperonsच्या मदतीने तो त्याची ॲक्शन दाखवू शकतो. Next one is HSP100. So it's known as CLP family of chaperones in case of E. coli or heat shock proteins in case of E. coli. They have been studied in vivo and vitro for their ability to target and unfold tagged and misfolded proteins. युबिक्विटिनने टॅक्ट प्रोटीन्स असतील तर त्यांना डिग्रेड करण्यासाठी आणि मिसफोल्डेड प्रोटीन्स असतील त्यांना प्रॉपरली फोल्ड करण्यासाठी HSP100 plays a significant role, and this activity has been studied in vivo as well as in vitro. Proteins in HSP100 or CLP family form large hexameric structure. So 6-unit structure is there, and they are involved in unfoldase activity in presence of ATP. They are responsible for removing the improper folding and stimulating the correct folding. These proteins function as chaperones by processively threading client proteins through a small 20 angstrom pore, thereby giving each client protein a second chance to fold. So client protein will be that protein which is not properly folded. So in that case, HSP100 will play the role of a molecular chaperone and it will give the second chance. It will remove the folding and it will stabilize that particular protein or client protein, and it will promote the proper folding of that type of client protein. चुकीची प्रोटीन फोल्डिंग असेल त्याला करेक्ट करण्यासाठी दुसरा चान्स देत असतो. Some of these HSP100 chaperones like CLPA, CLPX associate with double ring tetradecameric serine protease known as CLPP instead of catalyzing the refolding of client proteins. These complexes are responsible for target destruction of tagged and misfolded proteins. काही HSP100 जे आहेत ते प्रोटीएस सेरिन प्रोटीएस जे टेट्राडेकॅमेरिक असतो 10 युनिट्स आणि त्याचे 4 कॉम्बिनेशन्स असा जो प्रोटीएज आहे त्याच्याबरोबर तो अटॅच होतो आणि अशा प्रोटीएसच्या मदतीने तो त्या अनफोल्डेड प्रोटीन्सचा किंवा इनकरेक्टली फोल्डेड प्रोटीन्सचं डिस्ट्रक्शन करायचं काम करत असतो. So again, it will be carrying those proteins towards proteasome for destruction. HSP104 and HSP100 of Saccharomyces cerevisiae, they are also essential for propagation of many yeast prions. I told you yesterday, prions are resistant to the action of proteolysis by proteasome. अशा वेळी हे HSP104 and 100 Saccharomyces cerevisiae मध्ये त्या प्रियॉन्सच्या प्रपोगेशनमध्ये युजफुल असतात. Deletion of HSP104 gene results in the cells that are unable to propagate certain prions. So here we finish with the fourth unit. I'll send you the units of the fourth unit, notes of the fourth unit today, and we'll start the fifth unit from tomorrow. Okay? Any difficulty? Did you understand? Any difficulty? समजलं का? Did you understand? Priya, Sonali, understood? I told you yesterday, prions are resistant to the action of proteolysis by proteasome. अशा वेळी हे HSP 104 and 100 Saccharomyces cerevisiae मध्ये त्या prionsच्या propagation मध्ये useful असतात. So we'll stop here and continue the things tomorrow. Thank you. Bioinformaticsचा chapter आहे तुमचा last. So we'll start with the bioinformatics chapter and you keep on studying. Today I will send you the multiple choice questions also. So you solve those multiple choice questions also. Okay. Notes of fourth unit and MCQs will be sent today. Bye and thank you.